Hi, everyone, and welcome. I'm pleased to be your host for today's webinar. My name is Dana Jaber, and I am a market development manager and part of the global customer support team at Bionano. We are delighted to have you with us for the second session of our Bionano in Motion webinar series. This series will spotlight the latest and most impactful studies using optical genome mapping, or OGM, in hematological diseases and beyond. For today's webinar, we are delighted to introduce our distinguished speakers who will present on the following. Dr. Hélène Guermouche from the Institute of Medical Genetics at the University Hospital Center, or the CHU of Lille, which is in France. She will present her team's work on how optical genome mapping allows to decipher the genomic complexity of multiple myeloma. Our second speaker, Dr. Agnès Daudignon, also from CHU Lille, and she is the coordinator of the French OGM group that is associated to the GFCH. She will present how this group helped in the study and how OGM can be an efficient tool in multiple myeloma. She will also show us how this network is facilitating the deployment of OGM nationwide in different heme indications. And our third speaker, Dr. Hippolyte Guerineau from the Hospital Center of Versailles, also in France, will share how they extended this study using OGM on a large prospective cohort on multiple myeloma samples. Dr. Guermouche, Dr. Daudignon, and Dr. Guerineau, thank you so much for being with us today. The floor is yours.
Thank you, Dana. Hi everyone, my name is Hélène Guermouche, and I'm going to start this talk with our experience, the preliminary work on multiple myeloma using OGM. So today, the first step for cytogenetic evaluation of multiple myeloma is plasma cell sorting, which is really mandatory for this indication. You know that, especially using FISH, we have to deal with the problem of a limited number of cells available in the samples, and we are facing an increasing number of targets to find, thanks to the literature. So far, for multiple myeloma, we need a technique that can detect all copy number variants, as well as balanced structural variants, and if we can, mutations. And this with as few cells as possible. So what about OGM? For now, you know that OGM still requires at least 1 million cells. Our experience is that sometimes it is difficult to obtain these million cells, and to avoid this problem, we have the idea to mix plasma cells with the non-tumoral cells from the same sample to reach the million required. But this brings up a new question. It is, how far can we dilute the plasma cells with OGM and still keep a decent level of sensitivity? So the first step of our work was to find the optimal dilution. We perform a dilution range from 1 BEM samples, and the plasma cell was sorted and mixed with 1 pure fraction of plasma cells, which was analyzed through FISH, and this plasma cells was also mixed in 3 mixes, 50% mix, 25% and 12.5% of plasma cells within 3 different mixes, and we keep the pure negative fraction also. And all these samples were processed through OGM using RVA analysis. What about the result of FISH for this sample? We detected on the pure plasma cell fraction loss of TP53 in 19% of the cells, and also a translocation 1140 with an additional signal for CCND1 in 96% of the cells. There were no 1P or 1Q gain or loss. What about OGM? this is just to say that quality metrics were okay, and we all detected the translocation 1114 and the deletion TP53, which was a deletion of short arm of chromosome 17 here. And in fact, the extra signal for CCND1 was in fact a trisomy 11. So what about the results of the deletion range? You can see here the circus plot for structural variant detection for all the fraction we analyze. You can see that translocation 1114 was detected until fraction 12.5% of plasma cells. What about CNV detection? We can see that at least until fraction 50, we can detect all trisomy 11 and deletion of short arm of chromosome 17, but this was only manually detected because it was subclonal deletion of TP53. So with this result, we decided that up to deletion of 50% of plasma cells, we were able to detect all the critical SV, for here the translocation 1114, and to detect the CNV, the clonal CNV. So we performed OGM using this new approach of mixing 50% plasma cell in the negative fraction for each sample in 13 additional BM samples. And we performed OGM, RVR analysis, and de novo analysis for translocation 1114. I will come back to this later. We also perform FISH results on pure plasma cell fraction, and this is the result for all the samples. You can see we have the known driver rearrangement, you know, in multiple myeloma. And OGM confirmed 13 of the 15 anomalies identified by FISH. 2 were missed. The first one is a subclonal gain of 1q21, which was in fact subclonal. This is the reason why I think OGM did not highlight this gain. And another one is the atypical translocation 1114, and I will come back to this later too at the end of the speak. But OGM also highlighted many other anomalies with a mean of 72 events per samples, including critical and known drivers of multiple myeloma, especially chromatopsis pattern and translocation involving immunoglobulin genes. About chromatopsis, OGM has emerged as a major tool for detection of chromogenesis, but the subject will be developed by my colleagues Hippolyte later. About the translocation of immunoglobulin genes, we were able to detect all translocation involving the translocation 414, the translocation 1420, which are known in multiple myeloma, but also translocation involving MYC with a light chain of immunoglobulin, which is not really routinely tested in our lab. Also here, another translocation, rare translocation of CCND2, also not routinely tested, and the frequency of this translocation may be underestimated due to the fact that it's not tested in routine. And I will finish with the translocation 1114. We had 3 cases with translocation 1114 by FISH. For the 3 of them, 2 were detected by OGM, 1 with RVR and de novo pipelines, 1 with only the de novo pipelines, and the 3rd one not detected either by RVR or de novo pipelines. Interestingly, we processed these 3 samples through panel-based DNA sequencing to detect the translocation 1114, which is performed in France for protocolar samples. But we had the same result, meaning the third sample would not DNA sequencing did not detect translocation 1114 for the third case. So for this particular case, it was unusual, but we had the karyotype of the plasma cells, and this is the karyotype. You can see the translocation 1114 and other anomaly, which were detected perfectly by OGM, so monosomy 13 and monosomy 22, as well as an unbalanced translocation between X and 17. So this is not a problem of sensitivity due to a dilution. When we performed a FISH looking for IgH and CCND1 translocation, thanks to FISH on metaphases, we can see that this translocation was atypical with a signal only on derivative chromosome 14 and not on the chromosome 11 of the translocation. So this, in fact, was an unbalanced translocation, and maybe, you know, it's possible to have unusual breakpoints in multiple myeloma for translocation 1114, because there is a high variability in genomic position of the breakpoint for this translocation. And sometimes the translocation may be, the breakpoint may be very far from CCND1. In this work, we noticed that using RVI, most of the time, only derivative chromosome 11 was detected. So maybe this is the reason why using this pipeline, we had trouble to detect the translocation. And especially when this is possible, the chromosome 11 is lost, you can totally miss this translocation. But the good news is that guided assembly, this is very new and it's not in the paper, but guided assembly detected the translocation. Here you can see the translocation of the circus plot, and here the molecule of the sample with the translocation. So to conclude, this work is the proof of concept that OGM is able to detect stratifying anomalies as well as FISH and DNA sequencing. With our experience, we detect all clonal abnormalities in one single test, which is very important and crucial with the problem of the number of cells, which is limited. OGM also greatly improves informativeness compared to FISH, because this is whole genome evaluation. And OGM, due to this work, remains sensitive even when you dilute plasma cell in your samples up to 50% in our experience. So this work is published. This is the title of the paper, and there is many other information that I have no time to develop here, but I'm happy to share this work if you want. We also notice some limitation and optimization prospects with OGM. So of course, the results here must be confirmed on larger cohorts, and sometimes we have incomplete description for subclonal population. We missed one Q gain due to subclonal, but sometimes we are, you know, it's also possible to have trisomy or tetrasomy of this chromosome rearrangement. So it's this one limitation. And also, I did not mention about stratification in multiple myeloma regarding TP53 alterations, but the cutoff needs to be evaluated in OGM too, because it's still evaluated by FISH, in fact. But we really think that OGM can be integrate in a workflow for routine diagnostic for multiple myeloma, and we propose here a workflow after plasma cell sorting, we still require one fish for TP53 evaluation due to this cutoff I mentioned before, and we can perform a mix if required, if we had enough samples to perform on pure section. Of course, we will do on 100 plasma cell fraction. We can perform OGM, DNA extraction, and we can perform OGM. So, and if we want, DNA from OGM extraction can be used also, because we perform DNA sequencing on our OGM DNA extraction to realize TP53 sequencing or panel-based DNA sequencing, if you want. So now I want to thank everyone involved in this project, which are seated here, and especially the frog. And I will let Agnès talk about this after.
Okay, thank you, Hélène, for this very nice presentation. My name is Agnès Daudignon. I'm not going to speak very long, just make the connection between the two presentations and to introduce you. As Hélène told you, this study, previous study, was supported by frog members. It's only way. So what is a frog? Frog is a GFCH working group dedicated to OGM, created in January 2023 to organize the deployment of the OGM in France. For now, we are 69 biologists and engineers from 26 laboratories, 20 from France, 2 from Canada, 2 from Switzerland, and 2 from Belgium. Next slide, yes. For now, we have 6 working groups dealing with various topics of interest, such as method of validation, 2 groups from biological validation of myeloid and acute pathologies or lymphoid pathologies and myeloma, 1 group dealing with control quality, 1 group working on the financial aspect of the OGM, and the last group working on the specific chromogenesis, from chromogenesis, which has a ramification with a European group scale from a Spanish, German, Israel consortia. Next. So the subgroup 3, which with lymphoid pathology, is divided in 3 categories, 1 working on the CLL, 1 working on non-Hodgkin lymphoma, and 1 working on myeloma. In this group of myeloma, we work about the method of validation for the local workflow from specific bed from myeloma, and 2 times from trimester we meet to discuss difficult cases. So, wait, next diapositive slide. So the OGM is implemented in local laboratories in France in CH of Versailles, directed by Christine Terré and Hippolyte Guerineau, and in the Cancer Center Henri Becquerel of Rouen, directed by Dominique Pinter. In Switzerland, the optimization is in progress in Lausanne. In Belgium, the OGM in myeloma is implemented in the Hospital of Liège. And in Quebec, the OGM is implemented in Montreal. Next slide. In addition, and for your information, the GFCH have published in 2023 its own guideline, the management of malignant hemopathy, in a special issue of 13 publications covering the entire field of hematologic malignancy. Next slide. OGM is mentioned as a promising alternative technology in most hematologic malignancy, notably in the B and T ALL, in CLL and in myeloma. So I'd like to hand over to Hippolyte Guerineau, who will tell you about Versailles experiment involving OGM in multiple myeloma. Thank you.
Okay, so hello everyone. My name is Hippolyte Guerineau, and I'm going to present our experience with optical genome mapping of multiple myeloma at the Versailles Hospital. So OGM has been implemented routinely for 9 months for myeloma samples since September 2024. So first, I'd like to introduce you to our workflow. We collect 2 bone marrow samples of approximately 4 ml, 1 in EDTA and 1 in heparin. If plasma cell infiltration is less than 80%, we perform plasma cell isolation, otherwise we do not. Note that we may mix the EDTA sample with the heparinized one if necessary. Next, the cell count is performed. If there are fewer than 0.5 million cells, we do not perform OGM, but instead we run 3 FISH tests, FGFR3 EGH, TP53, and 1P, 1Q. If there are between 0.5 million and 1 million cells, we dilute the sample with negative fraction to achieve at least 50% of plasma cells. And if there are more than 1 million cells, we don't dilute with negative fraction. And if there are more than 2 million cells, we also prepare a backup sample. So finally, we extract and label the DNA. During the first 3 months, to validate the method, we performed OGM alongside FISH for TP53, 1P1Q, and IGH CCND1. We use both RVR and de novo assembly because 1114 translocation was not always apparent, even when all features were removed, as you can see here. We also use de novo assembly to assess ProID if necessary. We start using the guided assembly to compare it. So currently, we only perform OGM and FISH TP53 to evaluate the percentage of plasma cells with TP53 loss and to be able to detect minority clones. We normally use a guided assembly, except in cases with ProID issue, where we also rely on de novo assembly. We evaluate myeloma risk using the IFM score, which includes the loss of TP53 in over 55% of plasma cells. And we also use the EMWG 2023 criteria, not yet published, which includes loss of TP53 in over 20% of plasma cells. For analysis, we use the bed file created by the FROG, so specifically by the myeloma group. Each is analyzed by 2 operators, and only abnormalities with prognostic significance are reported. So after analysis, ultra-high molecular weight DNA is sent to molecular biology department for TP53 mutation testing, as it's part of the EMWG score. Now let's talk about our experience. So between October 2024 and March 2025, we received sample of 124 newly diagnosed myeloma, and 114 of these were successfully analyzed. Among the 113 samples analyzed, most underwent plasma cell isolation without dilution, meaning they had more than 1 million cells. And in terms of metrics, most of them were optimal, meaning a MAP rate over 70% and an effective coverage after analysis over 300. Now let's focus on the different abnormalities. So regarding IGH rearrangements, we found frequencies similar to those reported in literature. However, we observed difference with more 1114 translocation and fewer 414 translocation, which we can't fully explain, perhaps due to the small number of samples. Indeed, by April, we have already identified 3 samples with 414 translocation. We also observed a similar frequency of samples with hyperdiploidy, defined by the gain of odd-numbered chromosomes. Regarding chromosome 1 abnormalities, we observed a lower frequency of 1Q gain. Since 1Q gain is considered as a rather secondary abnormality, this may be explained by the difficulty in detecting of subclones. Several studies have shown that the prognosis of 1Q gain depends on clone size, typically using a 20% cutoff, though it remains debated. Even with the reported frequency, it's not always clear what clone size was considered for 1Q gain. For del1p32, we found similar frequencies to those reported in the literature. We also observe a similar frequency of MYC rearrangements, with the majority of the partners being chromosome 14 with IGH, chromosome 6, chromosome 1, and chromosome 22 with IGL. And rearrangements under MYC and IGL is reported as an adverse risk. In terms of risk stratification, we found frequencies similar to those described for the IFM score and for the EMWG 2023 score. It's worth noting that the number of high-risk samples according to the EMWG score is probably underestimated, as the TP53 mutation result was not always available. Now let's focus on TP53. So we detected TP53 loss in 10 samples. In 8 of these cases, the clone size was similar between OGM and FISH. Among those 8, 2 were only visible after removing all features. This involved the TP53 losses in approximately 20% of plasma cells. In over 2 cases, there are discrepancy in clone size, with FISH showing a larger clone than OGM. We're not entirely sure why this difference occurred, but in all cases, TP53 was lost in more than 50% of plasma cells, which correspond to a high-risk prognostic score in both prognostic scores. So in conclusion, we are still performing TP53 FISH because the clone size threshold in the prognostic score have not yet been validated using OGM alone. Even people who run NGS very often perform TP53 FISH. However, these results are very promising. Now, there are other more recent prognostic markers apparent thanks to OGM. So here is a study by Strong and his colleagues. They investigated the breakpoint on the derivative chromosome 4 in the 414 translocation. The finding shows that the prognosis varies depending on the breakpoint location. If the breakpoint is located before the NASD2 gene 414 translocation, is not considered as high risk. If it occurs at the start of the genes, the prognostic is considered intermediate. And if the breakpoint is within or beyond the middle of the genes, it's associated with a high risk and with a decrease of survival probability. Thanks to the bed file created by the FroGG myeloma group, we are able to evaluate these breakpoints more precisely. However, it's not always clear-cut, as some breakpoints may fall between 2 defined regions. Finally, complex structural variants, notably chromoanagenesis, are found in around 40% of samples. In myeloma, chromotrisis has been reported as an adverse risk factor. However, it has been defined using molecular biology techniques, such as whole genome sequencing. Therefore, further studies are required to better understand its role and significance in myeloma. So there is a group at FroGG whose role is to characterize these complex events. So here are a few examples with a circle plot that may suggest a chromotrisis of chromosome 13 and maybe chromosome 8. Here's one with that may suggest a chromoplexy. And here's a very complex structural variant with possibly several phenomena at the same time. So in conclusion, in multiple myeloma, OGM is concordant with FISH and provides additional information without multiple FISH tests, such as breakpoint analysis and chromoanagenesis. Only TP53 mutation testing is required to assess the risk of multiple myeloma according to recent prognostic score. So OGM is successfully implemented in the workflow for multiple myeloma. So thank you so much for your attention, and I'm happy to take any questions.
Thank you, Hippolyte. Thank you, Hélène. Thank you, Agnès, for this incredibly insightful presentation. As the title of the talk says, from need to reality, it's really exciting to see how OGM is uncovering those structural variants that were either undetectable or not well characterized in multiple myeloma, especially in multiple myeloma, where the need to improve the detection of these events is huge. Thank you, Hélène. It's really amazing to see how your team validated OGM in multiple myeloma and how you came up with a workaround for the samples with less amount of CD138 plus cells. Hippolyte, it was really nice also seeing how you shared the experience of the Versailles hospital, and you showed us that the workflow that Hélène and her team worked on is easily implementable in a lab setting. And thank you as well, Agnès, for showing us how the FroGG group is collaborating and helping the deployment of OGM in the different hospitals of France. Once again, a big thank you to our speakers and thank you to the attendees. Thank you for being a part of today's webinar. Have a wonderful day, and we look forward to see you in our next webinar. Thank you.
Thank
you.